Hello everyone, today we will discuss about the degeneration and the regeneration of the neurons. Now first of all, what is degeneration and regeneration? Degeneration, that means the degenerative changes which occur actually in the axon after it is injured. So what kind of injury things it will show, okay, what kind of degeneration occur in the axon after the injury, and what is regeneration, that means what kind of repairing it will do if possible. Now when the axons are actually injured, a series of degenerative changes are seen at mainly three levels of the axon. Suppose I consider that at over here somewhere the neuron is injured, so it will show degenerative things in the degenerative sites at three places, that is distal to the injury somewhere over here, then proximal to the injury, and at the cell body. Distal to the injury, proximal to the injury, and in the cell body. So the first of all, stage of degeneration. Now what is anterograde or the Wallerian degeneration? That means the degenerative changes which occur in the part of the axon distal to the site of the injury is referred to as anterograde. That means somewhere over here if the injury is present, so distal to that is known as a Wallerian degeneration. Okay, and proximal to that is known as the retrograde degeneration. That means the site of injury proximal to the axon somewhere over here is known as the retrograde degeneration, and the third one is the cell body degeneration. Now the Wallerian degeneration or the degeneration, that means actually or the degenerative changes which occur in the part of the axon distal to the site of the injury. Now it mainly starts within the few hours of injury and continues for about 3 months, means up till 3 months this Wallerian degeneration will continue. Now what happens actually in the axis cylinder? What happens, what changes occurs when the degenerative changes occur distal to the injury? So as you can see, this up till E we have degenerative process, and after that regeneration. So right now we are concerned with this part. So now over here the axis cylinder actually becomes swollen and irregular in shape within a few hours of injury. After a few days it breaks up into small fragments. As you can see over here, it will break up into small segments. And the neurofibrils, I told you what is neurofibrils in my nerve video. The neurofibrils are actually the filaments of the neurons, right? So they are the filaments, or we can say the microfilaments of the neurons. So the neurofibrils will actually break down into granular debris and are seen in the space occupied by the axis cylinder. As you can see, the small things are the neurofibrils debris. They are the parts or the fragments of the neurofibrils. So they will be seen all around in the axis because they will break up into fragments. Now the changes which actually occurs in the cell body. So now what happens over here means in this part, the soma or the perikaryon part, the changes in the cell body of the injured neuron actually starts within 48 hours and continue up to 15 to 20 days. While over here, the changes in the Wallerian degeneration actually continues, starts from within a few hours only. While over here, it will take around 48 hours to start and it will continue up to 15 to 20 days. Now changes actually occurs in the cell body are first of all the nasal substance. I told you what is nasal substance. It is actually the substance which are present in the cell body which actually continue to the dendrites also, but not up to the axon hillock or in the axons, right? So the nasal granules or the nasal substances are actually the made up of rough endoplasmic reticulum. So these substances undergo degeneration and dissolution, which is also known as the chromatolysis. Now the Golgi apparatus, mitochondria, and the neurofibrils also are fragmented and eventually they will disappear. And the cell body draws in more fluid, enlarges and becomes spherical. So this cell body will take up more fluid and become swollen. Nucleus, which is present at exactly the center of the cell body, it will be displaced to the peripheral part towards the cell membrane. And sometimes the nucleus is also extruded out of the cell, in which case the neuron atrophies and finally disappears completely. So if the nucleus is not present, nucleus is very important for the regeneration, the repairing part of the particular injured neuron, if the nucleus is displaced or extruded out of the neuron, then this neuron will actually atrophy and finally disappear completely, and it can't repair further. Now the stages of the regeneration or the repairing process of the injured neuron. Over here, this is the diagram of the repairing neuron. Now it begins at around 4th day and is more active after a month, and it may last for around months or years. Okay, it will take time to repair. Now the factors which actually affects the repairing process of the particular neurons are, first of all, just remember that crushed neuron actually gets easily repaired and faster repaired as compared to the cutted neurons. While the cutted neuron chances of regeneration or the repairing is increased if the cutted ends are close to each other, that means around 3 mm. If they will exceed than this, then their capacity for regeneration will actually decrease. Also, presence of neurolemma. What is neurolemma? It is the membrane of the neuron, is very important for this process. Now you may know that the CNS has neurons and the neurons of the CNS have no neurolemma. So that's why the CNS neurons, once degenerated, never regenerate, as the nerve fibers have no neurolemma. Now the presence of nucleus in the neuron cell body is also must for the regeneration process to occur. If it is extruded out, the neuron is atrophied and regeneration does not occur. So main two things, neurolemma and nucleus, is very important for a particular nerve or particular neuron to be get repaired. Now the regenerative changes or the repairing changes occurs in two ways, anatomically and functionally. In anatomical, the structure of the neuron actually repairs, while in functional repairing, there is the neuron actually gains its function back of transmitting the impulse. Okay, the anatomical actually is a faster one as compared to the functional. Functional will actually take time because the neuron is actually weak and they will take time to gain back its previous function. Now first of all, anatomical change or the repairing, it is done in two ways, change in the axon and the change in the cell body. The first of all, change in the axon has mainly 4 very important steps. That is, first of all, fiber formation. Now what happens is the fibers are actually around 100 to 200 fibers actually elongates and comes out from the proximal, remember, proximal part of the cutted end of the neuron, and they will extend towards the distal part of the cutted end of the neuron, and they will do effort to reach the distal cutted end. This is the first process of the formation of the fiber formation. And next is the stage 2, which is the entry of the fibrils into the endoneural tube. Over here what happens is the strands of the Schwann cells from the distal cut end of the axon actually guides the regenerating fibrils to enter the axon endoneural tube. So means in the first step, the proximal part was giving its fibrils to the distal part. Okay, but in the second stage, what happens is the Schwann cells of the distal cut ends will actually guides the fibrils of the proximal part to actually enter into their endoneural tube. And once it actually enters into the endoneural tube, they will show very fast growth. Now next is the active growth stage. Now over here, the axonal fibril growing through the endoneural tube enlarges and establish contact with the peripheral end organ. So means the fibril will actually associate with the peripheral end organ, and the new axon formed in this way is devoid of myelin sheath. Remember, over here up till stage 3 there is no myelination of the neurons, so means they are still weak. Okay, so up till stage 3 there is only reaching of the fibril up to the distal ends, entering into the endoneural tube by the help of Schwann cells, and then there is active growth which actually in which the fibrils actually makes a contact with the appropriate peripheral end organ. Now up till this process actually takes time for about 3 months. Okay, so you can imagine how difficult this process is, and it will take a lot of time for about 3 months to reach up to stage 3. Now stage 4 is the myelination. The myelin sheath is then formed slowly by the cells of the Schwann or the Schwann cells, and the myelination is completed in around 1 year. So means this stage occurs in 3 months, while this stage requires around 1 year. Okay, so regeneration has 4 main steps of the axon: fiber formation, enter fibrils into endoneural tube by the help of the Schwann cells, active growth in which they make a contact with their peripheral end organ, and then the myelination, very important step, which actually takes 1 year around. Now the changes occurring in the cell body for repairing. Now over here in the perikaryon, I told you that it has nasal granules which are made up of rough endoplasmic reticulum. The nasal granules followed by the Golgi apparatus appears in the cell body. Before this, in the damaging or the degenerating part, the nasal granules were actually degenerated and almost disappeared, and Golgi body also was disappeared, right? But they will appear again in this repairing process. The cell in this process will actually lose excess of the fluid and regain into its normal size. In the degenerating process, it was actually taking a lot of fluid and actually it was swelled up. Okay, but over here it will not take any further water, but it will lose excess of the fluid and regains its normal size. The nucleus, which was actually displaced in the degenerating part or the degenerating process, it will actually occupy the central position and will come into the center of the perikaryon. Now all these changes will start within the 20 days of the injury and completed in around 80 days. Now the functional regeneration process of the neuron, injured neuron. Over here I told you before that the functional regeneration actually takes time, because as we all know, main function of the neuron is to transmit the impulses, right? So it will take time because the neuron actually are very sensitive. So once they are degenerated, it will take time to regenerate and to repair. So it occurs after a very long time. Now next is the factors that promote the neuronal growth. Now various factors affecting the neuronal development, growth and survival have been isolated and studied. So these are mainly two: neurotropins and the other factors that affects the neuronal growth. Now first of all, the neurotropins. Okay, now these are the proteins actually which provide the trophic support to the neurons. What is trophic support? That means they promote the nerve growth and the survival of that particular nerve. They also have a role in neuronal plasticity. Now they are mainly produced by mainly the muscles of the body and other structure also, especially by the gland that the neuron innervate. Okay, means they are mainly formed by the muscles and the glands which actually are innervated by the nerves. Just remember this, by the neurons. Some of the neurotropins are actually produced by the astrocytes also. So means muscles and the glands which actually innervated by the neurons will produce, and the astrocytes also produce the neurotropins. Now established neurotropins include the following three parts or the three types. That means the NGF. What is NGF? Nerve growth factor. BDNF, that means the brain derived neurotropic factor. And third is the NT3 and 4/5, that is neurotropin 3 and 4/5. Now first of all, the nerve growth factor. It is mainly the first neurotropin recognized, and it promotes the growth of the sympathetic nerves and some sensory nerves. Just remember this. Promotes the growth of the sympathetic nerves and the sensory nerves. Now they are mostly found in various tissue in the human being body and many species of the animals, but they are found in high concentration in the submaxillary salivary gland of the male mice. Just remember, the submaxillary salivary gland of the male mice has a high concentration of this NGF or the nerve growth factor. Now the NGF is actually made up of 2 alpha, 2 beta and 2 gamma subunits. Okay, now this 2 alpha subunit actually, or the alpha subunit, have trypsin-like activity. The beta subunit are similar to the structure of the insulin and possesses all the nerve growth promoting activities. So means this for it has mainly trypsin-like activity, insulin-like activity, and also the nerve growth promoting activities. And the gamma is actually nothing but a serine protease. Okay, and its function is not known yet. Now the receptor of the NGF is the TRKA. The full form of this is tyrosine kinase activity A. The receptor of NGF is tyrosine kinase activity A. Now BDNF, brain derived neurotropic factor. Now this has mainly growth promoting role for peripheral sensory nerves. This was for sympathetic nerve sensory nerve. This is for peripheral sensory nerves. And last is the neurotropin 3 and 4/5. Now they are mainly promoting the role for growth for cutaneous mechanoreceptor. So they mainly do the process for the growth of the cutaneous mechanoreceptor. So neurotrophins of 3 types: NGF, BDNF, NTC and 4x5. Now next is the other growth promoting factors are mainly 4. Just remember the names: insulin-like growth factor 1, fibroblast growth factor, transforming growth factor, and the PDGF. This is the platelet derived growth factor. So fibroblast growth factor, insulin growth factor 1, transforming growth factor, and the PDGF, platelet derived growth factor. So this was all about the degeneration and the regeneration of an injured neuron. I have made one separate video on the particular nerve in which you will get the full information about the neurons and its types, subtypes and all its activities and the constituents. Just watch that first, then only you will be able to understand about this video much. Hope you understood well. Thank you so much.